So is my okay, let me share my screen one more time. How was your day? Good. So this is our last class and we have to complete the paper of 2021 for the like very most important thing first of all, and then you can ask whatever like is confusing part for you. Okay, let's start with question number 6. We are done with 5 questions. Okay, so here we have a graph, and the figure 7 shows the mean distance between centromere and the poles of the spindle during the mitosis. Calculate the rate of movement of centromere during phase E. So this is phase E starting from here and ending here. Okay, and we need to give answer in micrometer per minute. We need to divide these to these three decimal places. First of all, we need to um, tell the initial value and then the final value and how much distance is moved. So what is the initial value? It's 19, like 19 micrometer. Let's say 19 micrometer at what seconds? Like 1800, around 1800. Is it so? Okay. So we have 19 micrometer at 1800, let's say 1800 seconds. And what is our final value? It would be around 4. Okay. At what? 4 micrometer at what second? 2500 second. Now what we need to do, we need to calculate the time first of all. How many seconds are there between 2500 to 1800? 700 seconds. And how many or like what is the difference in the distance, like 19 minus 4? 15. So it moved 15 micrometer in 700 seconds. So we need to divide this. Yes. 0.0214 micrometers in this second. Is it so? So calculate. Okay, now the rate is actually the micrometer. So how we calculate the rate for like the movement? Have you done the transpiration thing, in which the bubble move and we need to calculate the distance moved by the bubble per minute? No, really? It's in the like O levels, not in A levels. Not a practicable but a part of ATP. No, okay. So whenever we want to calculate the rate, we find the distance over time. So the distance over time, oh sorry. So the distance over time is actually in seconds is 0.0214. Now how to convert this into minutes? Convert it into minutes because they have asked us about the conversion in minutes. Have you taken physics? No. Okay. So we say the rate of reaction or anything, the rate is distance divided by time. Okay, this is the general formula that we use. So we have taken the distance, we have taken the time. Now we need to follow the instructions. We have time in minutes. Can you convert 700 seconds into minutes? Yes. 11 point 11 point is 18 and it's okay 1100. Oh, sorry, 11.666. So 15 divided by 11.666. So it would become 1.29 if we round 23 decimal a 1286. Okay. So the answer is 1.286. So we need to round it off. Done? Okay, let's move to the next question then. Let me just copy the question. Okay, name three phases of mitosis shown by C, D, and E. So, what do you think? What is C? What is D? And what is E? First of all, we need to name these. Okay, okay. E is the Anaphase because the distance is decreasing, the spindles are moving towards the opposite ends. That's why the distance is decreasing. So in the prophase, the spindle fiber is in the formation form, like it's not properly, you can say, arranged, but the distance is, if we draw a diagram, it will be like this. Let's say now in the prophase, the chromosomes might be here, and in anaphase, the distance is maximum. Why? Because they arrange themselves on the equator. Oh, sorry, metaphase. In the metaphase, they arrange themselves on the equator, so the distance is maximum at metaphase. And in anaphase, when they are pulled apart towards their poles or towards the opposite ends, the distance reduces. Okay, so let's describe the role of spindle fiber and the behavior of chromosome during each of this phase. What happens in prophase? The chromosome formation takes place. The DNA condenses into chromosome. Okay, okay. So correct. Correct. So in prophase, it started to form. The centrioles move to the opposite end and they start to form a spindle fiber. In metaphase, the centromere attached to the spindle fiber and align them. Sorry, in prophase, they attach to the spindle fiber, but in metaphase, they align themselves on the equator. And in anaphase, the centromere divides because they are pulled apart from the center, and the spindle fiber shorter because they are retracted toward their opposite ends. Correct. So the next thing, ACD are the molecules. Let me just copy this question. Okay. ACD are the molecules that made of a monoclonal antibody linked to a cancer drug. Figure 8 shows how an ADC enters and kills a tumor cell. The process of entering the cell and breaking down of antibody to release a drug is very simple to phagocytosis. Use your knowledge of phagocytosis to describe how an ADC enters and kills the tumor cell. First of all, how do okay, okay. What's the role of lysosomes? Okay, lysosomes. So we have to write about lysozymes and then what is the role of lysozymes. Correct. So these three marks would be based on these three things. Now the next question. Sorry. Some of the antigen found on the surface of tumor cell are also found on the surface of healthy human cell. Use this information to explain why treatment with an ADC often causes side effects. Here is a question. Okay, wait. ADC binds to healthy cells. Okay, causes them to damage. Okay, so the tissue and an organ or a system can be damaged. Can be damaged. Correct. It's just like an autoimmune, in which the healthy white blood cells are going to be killed or eaten. So the next question. We don't have an image, so we cannot uh, work on this paper on this question. Okay, so we cannot do this calculation as well. Let me just go through the question and let's see whether we'll be able to solve it or not. So the question says that we have, what do we have? Mice in group H were injected with 2 milligram per kg of molecular antibody, and the antibody was in the solution of the concentration. This calculate the volume of antibody solution the scientists would would have injected into a 23-gram mouse. Give your answer in cubic decimeter. Antibody solution. So we need to calculate the volume. Uh, Logically, we can calculate uh, by calculating with the help of formula of concentration. Okay, let's do it. Let me first copy the question. We can do this. Okay, now how do we define a concentration? Concentration is actually the mass in a given solution or the moles in given solution. Here they have given us mass. So let's say the concentration or is the mass in a certain volume. Okay, it might be gram per cubic decimeter. For example, here they have given it milligram per cubic decimeter. This is the concentration they have given. Is it so? Okay. So we can use the same formula to find it, but first of all, we need to find the mass of antibody. Now, how to calculate the mass of antibody? So here they have given us that 2 milligram of and per kg of antibody is given to group H, okay, and then we have a concentration of 500. So first of all, let's convert this, the mass of antibody with the mass of mouse, which is this. So it would be 2 into into okay. Here we have grams. Now I'm telling you the answer. How would we do it? We have per kg, right? And this unit is in grams. Units should be same. So first of all, we convert grams into kg. So 23 means divided by 1000. It would be 0.023. So 2 into 0.023 is the mass of antibody, which would be, I get 46 and because 46 and 2 decimal places, so it would be 0.046. Okay, mg. Okay, the mass is 0.046. The volume given here is 500. So what could be the answer? Yes. Yes. Okay, correct. Suggest one reason. Okay, we cannot say this reason because the graph is not given, so we cannot say what is given in the mice. But why do you think the data cannot be given after 8 days? Uh, what else can be? Like they may have in the graph they may have the data till day 5. Why not after day 8? Yes, because of these, maybe the antibodies are not that much effective to protect them, so they may die. So we have done this a lot. So I'll tell you my experience that when I was like doing my MPhil, so we have to what was my topic? My topic was to, because at that time the dengue fever was quite the recent one, just like the COVID. So at that time dengue fever was the recent one. So we were given one thing that we need to find what how the different medicines can be used to check the effectiveness. So we performed experiments on mice, not on mice, sorry, rabbits. We were successful in saving them and in finding out a solution of how a certain medicine can be effective against something. Because we are chemists, so we were like, I'm a biochemist, so we have to deal with the microorganisms or the living organisms and the medicines.